Okay, hello everyone. I want to start giving just a few words about to give a little of a background of what we're doing in Human Molecular Genetics Lab, and then to introduce the work we do. First of all, in our lab, we work with primary cilia biology, studying how it is formed and how it's functioned. And primary cilia are organelles that are present almost within every cell in the body. And it hosts a lot of receptors and signaling pathways associated with these receptors that influence a lot of processes within the cells and the organisms, such as differentiation, metabolism, and homeostasis. The patients that present the mutations that affect the genes related to formation or function of primary cilia and generate a bunch of diseases that are called in altogether ciliopathies. Of that, among them, the one that we are studying at the Human Molecular Genetics Lab is the Vanderbilt syndrome. And one of the manifestations in this syndrome is obesity. So we think is that if we study how obesity is developed in these syndromes, we can give also some information on non-syndromic obesity, such as common obesity and the complications that may arise from it. So if we talk about obesity, we're talking about in part of white adipose tissue, that is the site of accumulation of energy within the body, in the form of neutral lipids. And it is also a site of hormone secretion and have other functions like insulation and mechanical resistance. During obesity, the white adipose tissue expands, but it can expand in different ways, okay? If there is an increase in cell number and an increase in differentiation, then the tissue expands in a more healthy way because the fat is stored in a higher number of cells. But in contrast, if the fat is accumulating within the cells that are already present in the tissue, the cells begin to expand, and this may affect their function and also may lead to cell death with inflammation of the tissue and dysfunction of the tissue. And this is thought to give rise to the complications, for example, type 2 diabetes and cardiovascular disease. So we are not studying the process of adipogenesis, and we think if we can know how it is regulated, maybe we can modulate it and shift the process of fat accumulation towards a more healthy setup. Okay, so adipogenesis is the process of generating of new adipocytes from the generation of new precursors, and also their differentiation. Okay, the differentiation of adipocytes involves the accumulation of fat first in different small lipid droplets within the cytoplasm, but then coalesce or join together and form a unique lipid droplet within the cell. In particular, we are interested in the study of the pathway that I mentioned in the bottom part of the slide, that is related to primary cilia biology. Okay, so in our lab, this process is studied mainly in cell tissue, cell culture, and also using mice models. And during my postdoc, my idea was to introduce the use of zebrafish model to study adipogenesis, as was mentioned before during the workshop. Zebrafish is a wonderful model to study in vivo processes because it's transparency. So you can put it in a microscope stage and watch things going on within the tissue. Zebrafish has white adipose tissue. It is developed during larval stages. And here we see an image of adipose tissue being labeled with night red. What we see here is the accumulation of fat. Okay. And also wanted to mention that white adipose tissue in zebrafish has almost the same characteristics as in mammals, okay? The distribution, it accumulates in several depots along the body, and also has similarities from the ultrastructural point of view and also from the gene expression point of view. So one of the problems before when I started to work with this is that there were not fish lines developed to label adipocytes. All the visualization of adipose tissue relied on lipophilic dyes. So we're looking just at fat, not the whole cell. So we weren't able to see earlier stages of development where there was not accumulation of fat. Okay, so the idea was first to develop a strain, a new fish line, to label the cell membrane of the adipocytes. Okay, so here is where Florencia Levin started to work with us. She was during her grade degree, she cloned several promoters from genes expressed in adipose tissue. Here I show you the one that finally worked out. So we are not working with a fish line that has the promoter from this transporter. This is a lipidic transporter expressed in adipocytes that direct expression of membrane EGFP. Okay, so here we see some images. Here are some larvae stained with lipophilic dyes in magenta, and in green we can see that the EGFP label is surrounding mature adipocytes within the lab. So then we take a look in more detail to this larva, and we saw that EGFP also labels some cells that didn't have lipophilic staining. So we think of these cells of adipocyte precursors. And it's also interesting that these cells have protrusions, and also we can see them moving around, okay? And also we saw cells that have several lipidic droplets, okay, suggesting an intermediate stage of differentiation. So then we try to figure out how we can use this fish line to study the process of differentiation in vivo. And here is where Leonel came into stage. Then he suggested that we can use NIRED and its spectral characteristics to study adipogenesis in vivo. As was mentioned before, NIRED is a lipophilic dye that will stain all the membranes in the cell, all the lipids in the cell. But it has the property that it is sensitive to the surroundings polarity. Okay, so if we put NIRED in a more polar environment, the spectrum is shifted towards the red. Okay, here we see it in vivo in cultured cells. Here are some cells that have lipidic droplets within the cytoplasm. If we surround a region with lipid droplets, here in one, we can see the spectrum of this region is blue shifted. And if we take a look at a region without lipid droplets, we can see that the spectrum is more red shifted. So maybe we can use this property to analyze the progression of adipogenesis and the accumulation of neutral lipids. So the experiment was like this. We stained larvae of different stages with NIRED. Then we anesthetize them and mount in agarose and do some confocal imaging. Then we do a lambda scan in the microscope and analyze the images using CMFCS and spectral phasors. Okay. The expected result will be that we will have 3 components, 2 due to NIRED and an extra one due to EGFP. Okay, and as was mentioned before also in the workshop, the pixels that lie within this triangle will have a mixture of the three components, okay? Here I show you, we first analyzed the individual components. And here is, there are some images. We first analyzed wild type larva staying with night red. So we get a cloud of pixels in this region that follow a linear distribution, meaning that we have a combination of that these pixels within this line will have a combination of more neutral lipids and more polar lipids. And if we analyze only the larva from the fish line, we're gonna have the cells will give rise to pixels within this region. So we have our three components well separated in the phasor plot. So now we turn to analyze larva from the fish line stained with night red. Here are some images. And one of the things that, okay. So one of the things that this allowed us was to segment the cells that express EGFP. It's because night red will stain all the cells in the tissue. So the EGFP label will allow us to individualize the adipocytes that are being during the differentiation and analyze only the night red profile in those cells. So here I show you already phasor plots from those cells. Those are isolated cells. And you may notice that the night red profile is quite different. So to be able to compare the profile among cells, we, and because we only wanted to analyze the night red profile, we just projected the pixels along this axis, the night red axis, and generated the graphs we are looking at in the bottom part, where we can see the percentage of pixels, okay, in the different levels of neutral lipid, let's say, okay. Then to analyze and compare between cells, we defined a center of mass for these distributions and also arrange. And in that way, we can compare among cells in larvae of different stages. Here we see other cells with different accumulation of, all of them have EGFP, but we also see that they vary in the night red, in the amount of night red and the profile of night red among them. And here we see the crude profiles. And in the bottom part, we are looking at the plot of the distribution range versus the center of mass of that distribution. So for different ages from different larval stages. So if we see here for the red dots, this is an early larval stages, so we will expect to find adipocytes in earliest stage of differentiation. And here we see that these cells have center of mass that are shifted towards lower contents of neutral lipids, so more polar lipid profile, and distribution more tight distribution ranges. But when we look at larvae from older stages, we are beginning to see distributions that are broader, okay? So we have a mixture of polar lipids and neutral lipids, and some cells that have more narrow distributions and a high content of neutral lipids. So this showed that this is a nice tool to analyze and quantify how the cells pass through the differentiation process in vivo. Luckily, all these results and some other, we could publish them this year. And how can we use this tool in our work to analyze this pathway? We are not, this is the work of, are being done by Lucia and Iliana, that are PhD students in our lab. And they already generated or are generating these mutants in fish. So we expect to analyze the differentiation of adipocytes in vivo within these mutants, just crossing lines. And also we can analyze the behavior of these cells in different diets, okay? If we, here is just a graphic showing that when we put the larva on high-fat diets, then we see a shift in the cells have more neutral lipid profiles shifted toward neutral lipids and more narrower profile. So we can analyze these two using this method. So to